Hello students, welcome to EPG Pathshala. I am Dr. Bibhu Prasad Panda from School of Pharmaceutical Education and Research, Jamia Hamdard, New Delhi. Here we are going to discuss about module determination of antimicrobials and disinfectants from paper Pharmaceuticals and Biological Analysis. After completing this module, you shall be able to understand what are antimicrobial substances, death of microorganisms, microbiological evaluation of antimicrobials, procedure involved in the evaluation of antimicrobials or disinfectants, and minimum inhibitory concentrations of antimicrobials and test for new disinfectants. Definitions of antimicrobials. The antimicrobial substances includes germicide, disinfectants, antiseptics, inhibitors, bacteriostats or fungistats or virustat, sanitizer and antimetabolites. Germicides are also called bactericides, fungicides or virucides. Germicides are the chemical substances which is used to kill bacteria or fungi or virus. When it is used to kill bacteria, it is called bactericides. When it is used to kill fungi, it is called fungicides, and whereas when it is used to kill viruses, it is called virucides. The disinfectants are the chemicals which is used to destroy the biological agents, that means the bacterial cells or the microbial cells that cause disease in man, animal or plants. Antiseptics are the chemicals which is used to prevent sepsis, that is blood poisoning, and applied to the tissues of human or animal to inhibit or destroy the invading microorganisms. Whereas the inhibitors are the substances that cause the microorganisms to grow at a slower rate than the normal rate. Bacteriostat, fungistat or the virustat, these substances that hold the number of bacteria, fungi or virus at a constant level. The sanitizer are the chemical that reduces the number of microorganisms and destroy any that might cause disease in human or economically useful animal. Antimetabolites are the substance that oppose the action of metabolites, that means the primary metabolites or secondary metabolites, and or prevent its assimilation in a microbial cell. Examples of antimicrobials: germicides. Under germicides, 8% formaldehyde, 2% glutaraldehyde, 100% ethylene oxide gas, and 10 to 20% hydrogen peroxides. Disinfectants. Under this, phenol, O-phenylphenol, chloroxinol, ethanol, isopropanol, iodine, chlorine, and benzalkonium chloride. Antiseptics. Under these, hydrogen peroxide 6% and iodine. Sanitizer. Under this, sodium hypochlorite, chlorine dioxide and peroxyacetic acid, that is PAA. Death of a microorganisms. Death of a microorganisms means inability of a microbial cell to produce growth within a given time in a defined media or medium at a defined temperature. What are the factors that influence death of a microorganisms? Number 1, viability of the microbial cells. Number 2, presence of inhibitor in the growth medium, that is presence of different type of antimicrobial substances. Number 3, the inoculum size or number of microbial cell, or it is also sometimes known as seed volume. Number 4, sensitivity of the medium. Number 5, different culture conditions or incubation parameters like incubation temperature, oxygen level, pH of the media and humidity level. Incubation temperature is an important parameter which influences the microbial growth. When a bacteria grown at lower temperature, the viability of the cells or the growth of the bacterial cells will be minimum. But when the bacterial cells were grown at around 35 to 37 degrees centigrade, the bacteria grow faster. Similarly, the oxygen level also very important here. If the oxygen levels are in good, the bacterial growth or microbial growth will be maximum for aerobic cultures, and for anaerobic cultures the oxygen levels or oxygen demand is required at a lower level. Similarly, the pH of the media also influence the growth of the bacteria and the microbes. If pH is kept at 7.2, the bacteria grow faster, and at a lower pH, for example at 5.5, the fungus or the yeast grow better. Humidity level also plays important role in microbial growth. Last factor, that is incubation time. Incubation time is also very important here. If the bacterial cells or microbial cells were incubated for a proper period of time, the bacterial cells grow faster, otherwise the growth will be minimum. Microbial growth analysis. The microbial growth analysis can be carried out by biomass. Mostly when fungal growth is analyzed, the biomass is measured. Respiration, that is the oxygen level or carbon dioxide level, also indicator for microbial growth. The dissolved oxygen level always reduces when there is a heavy microbial growth, and vice versa, that is CO2 level, that is carbon dioxide level, also increase when there is a heavy growth of the microbial cells. The metabolite determinations, the concentration of the metabolites, that is primary metabolites and secondary metabolites, also indicate the microbial growth. Optical density of the microbial cells at 620 nanometer is also indicator for microbial growth. Higher the optical density, more is the microbial growth, whereas lower optical density means there is a lower microbial growth, which is measured by using nephelometer or by using a spectrophotometer. Sometimes the microbial growth also analyzed by visual inspection. Why microbiological evaluations of antimicrobials is important? Number 1, to estimate the potency of the antimicrobials. The difference in the activity of an active and inactive form of chemical can be determined only by biological assay. The activity of the preparation is compared to the activity of a standard preparations. Number 2, to obtain the phenol coefficient. Determination of the concentration of a disinfectants required to produce a particular result compared to the activity of pure phenol that gives the same result. To confirm that the antimicrobials are active at the concentrations recommended for use, also called use dilution test, the performance of the test disinfectants is compared to that of hypochlorite. To determine the efficacy and limitations of new antimicrobials or antimicrobial substances, nowadays, before marketing of any new antimicrobial substances, the manufacturer carry out a series of proving tests that include the phenol coefficient test, the minimum inhibitory concentration for range of microorganisms, the effect of organic matter, and acute and chronic toxicity. Method of evaluation of disinfectants or antimicrobials. Phenol coefficient test. There are different type of phenol coefficient test. Number 1, suspension test, and number 2, surface film test. Under suspension test, number 1, the radial Walker test and the Chick-Martin test. Under surface film test, use dilution test and carrier test. Next method of evaluation is Nephelometric method, then capacity tests and minimum inhibitory concentration. Phenol coefficient test. The minimum concentration of pure phenol that produces viable subcultures at one time of sampling, but non-viable subculture at the next is compared to the minimum concentration of test disinfectant that produces same result. The test parameters are microbial strain, culture condition, microbial numbers, the temperature and expose time, the sample size, the time and temperature of incubation are fixed arbitrarily as part of test specification. The phenol coefficient test is performed by suspension test and surface film test. Suspension test. This is also known as the radial Walker test. Radial Walker test is invented in the year 1903 and it is applied in the year 1934. The strain used in radial Walker test is Salmonella typhi. Salmonella typhi is cultured for 24 hours at a temperature of 20 degrees centigrade. The phenol is diluted from 1:95 to 1:115 and the test substances or the disinfectant is diluted from 1:400 to 1:800. Subcultures are performed from both the test and phenol at interval of 2.5, 5, 7.5 and 10 minutes, and the plates is incubated for 48 to 72 hours at 37 degrees centigrade. The observations were made as growth or no growth. The dilutions of disinfectants which disinfected the suspension in a given time is divided by that dilution of phenol which disinfectants the suspension in same times gives its phenol coefficient. Now, how the disinfectants were tested or how the phenol coefficient test were carried out. Now you can see the table that the test disinfectant, the dilution number 1:600 shows a similar pattern of growth when it is compared with phenol at 1:100 dilutions. That means at 2.5 minutes it shows growth, at 5 minutes it shows growth, at 7.5 minutes it shows no growth, and at 10 minutes it shows no growth. The phenol coefficient is calculated like this: the dilutions of test disinfectants divided by dilution of the phenol, and it gives 6. That means the disinfectant's phenol coefficient is 6. Disadvantages of radial Walker test. In radial Walker test, there is no organic matter is included. The microorganism, that is Salmonella typhi, may not be appropriate since it is not very resistant to disinfectants and not a common pathogen nowadays, and can be replaced with Staphylococcus aureus, which is nowadays preferred. Time allowed for disinfection is short, used to evaluate phenolic type disinfectants only. Chick-Martin test. The microorganism used in Chick-Martin test is Salmonella typhi and or Staphylococcus aureus plus dried yeast. Dried yeast is added as an organic load. The temperature to perform the experiment is 30 degrees centigrade. The recovery media after contact time of 30 minutes, and the loop full of culture is incubated in a recovery media for 48 hours at 37 degrees centigrade. The observations were made: growth or no growth. The phenol coefficient, that is Chick-Martin phenol coefficient, is equal to mean of the lowest concentration of phenol prevent growth in subculture divided by unknown disinfectants. The phenol coefficient is lower than that given by Radel-Walker test. That means the phenol coefficient obtained by Chick-Martin test, the phenol coefficient is lower than that given by Radel-Walker test. When you compare both the method, that is Radel-Walker and Chick-Martin, you can see the volume of the media used in the Radel-Walker is 5 ml, whereas in Chick-Martin it is 10 ml. Diluent for disinfectants, in Radel-Walker it is distilled water, but in case of Chick-Martin it is water with yeast suspension. The yeast suspension or fecal matter is used as a organic matter in Chick-Martin test. The reaction temperature for Radel-Walker is 17.5 and for Chick-Martin it is 30 degree centigrade. That means the contact between the disinfectants and the microbe is carried out at 17 degree centigrade for Radel-Walker test and 30 degree centigrade for Chick-Martin test. The microbial cells used in Radel-Walker test is Salmonella typhi, whereas for Chick-Martin it is Salmonella typhi and Staphylococcus aureus, or Salmonella typhi or Staphylococcus aureus. Sampling times for Radel-Walker test is carried out after 2.5 minutes, 5 minutes, 7.5 minutes and 10 minutes interval, whereas for Chick-Martin test only one sampling is carried out, that is after 30 minutes of reaction. The calculation of the phenol coefficient is for Radel-Walker is carried out by dilution test killing in 7.5 minutes divided by same for phenol, whereas for Chick-Martin the mean concentration of phenol showing no growth after 30 minutes divided by same for test. Chick-Martin test calculation. Now you can see the table. The phenol percent, that is 1.8, giving no growth, that means showing a negative, and the phenol percent when it is 1.45, it's showing a growth in both the tubes, whereas in test disinfectants the percentage, that is 0.41, showing no growth in both the tubes, and at a concentration of 0.37 it shows growth in both the tubes. That means phenol coefficient can be calculated as shown in the slide. You can see there is a mean is calculated, the mean of both is calculated. That means the phenol coefficient is calculated by dividing the phenol percentage and the test disinfectants percentage, which gives 4.15. Surface film test use dilution test. The film of bacteria dried on a suitable surface with organic substance like protein substance. The test microorganism used are Staphylococcus aureus and Pseudomonas aeruginosa. Carrier, that is stainless steel cylinders, are meticulously cleaned, sterilized by autoclaving in a solution of asparagine, cooled and inoculated with a test microorganism by immersing in one of the culture suspension. The cylinders are drained on filter papers, dried at 37 degrees centigrade for 40 minutes, exposed to the used dilutions of disinfectants for 10 minutes, and cultured to assess the survival of the bacteria. Used dilution test is performed to confirm the efficiency of disinfectants dilutions derived from phenol coefficient test. Generally, 20 times the phenol coefficient is satisfactory. If the phenol coefficient of a disinfectant is 4, then use dilution test. The dilutions of disinfectants could be 1 in 4 multiplication 20, that is equal to 1 in 80. Carrier test, or it is also known as robot Koch method. The principle behind carrier test is a carrier such as silk or catgut or a penny cylinder, that is a small stick or a little stick, is contaminated knowingly by a microorganisms. After that, the carrier is dried and brought in contact with the disinfectants for a given exposure time and cultured in a nutrient broth. If no growth is there, that means the disinfectants is pass, whereas the growth indicates a failing. Limitations of carrier test are: the number of bacteria dried on a carrier is hard to standardize. The survival of bacteria on the carrier during the drying is not constant. The disinfectants residues were also carried over to subculture medium. Nephelometric method. It is first proposed by Needham in the year 1947. In the nephelometric method, the nephelometer is used for analysis of survival of Salmonella typhi after exposed to disinfectants. The objective of the nephelometric method is the damaged and untreated microorganisms may not reproduce at the same rate. The treated samples were incubated for 5 hours at 37 degrees centigrade, whereas the untreated samples, that is 3 number, also incubated for 5 hours at 37 degrees centigrade. After the incubation, the optical density or the opacity of the culture media is measured by nephelometer. The results of untreated microorganisms, the reference curve is plotted and used for calculating the number of microorganisms in the treated tubes. The advantage of this method is short incubation time, that is 5 hours, whereas the limitations of this method, that is very sensitive. This method is very sensitive. Capacity test or Kelsey-Sykes test. The principle behind the capacity test is the ability to retain activity in the presence of an increasing load, that is increasing microbial load, is the capacity of the disinfectants. In a capacity test, the disinfectants is challenged repeatedly by successive addition of bacterial suspension until its capacity to kill has been exhausted. The microbes used in capacity test are Staphylococcus aureus, E. coli, Pseudomonas aeruginosa, and Proteus vulgaris. The 3 successive loads of bacteria, that is addition of bacteria, carried out at 0 minute, at 10 minute, and at 20 minute. The temperature of reaction is kept at 20 degrees centigrade. Instead of loop, the calibrated pipettes are used for subculture purpose. Both the conditions, that is clean and dirty conditions, were maintained during the capacity test. The assessment of the test is carried out based on kill or not kill, but not by phenol coefficient. Now we can discuss how the capacity test is carried out. A bacterial suspension of 10 ml is taken and a disinfectant suspension of 3 ml is taken, and the bacterial suspension, that means the 1 ml of bacterial suspension, is transferred disinfectant solution at an interval of 0, 10 and 20 minutes. After 8 minutes, from the disinfectant tube, 0.02 ml of the microbial inoculum is recultured in other tubes of 5 number, and after 18 minutes 0.02 ml of disinfectants, after 18 minutes 0.02 ml of microbial inoculums were transferred again into 5 new culture tubes, and after 28 minutes 0.02 ml of microbial inoculums were again transferred into 5 test tubes, and all the test tubes are incubated for 48 hours at 32 degree centigrade. After the incubation is over, the capacity test table were made. You can see here concentration of the disinfectants are 1.0, 1.5 and 2, and the inoculum counts were kept constant at 2 into 10 to the 9 cells per ml of the inoculum volume. And in the challenge number 1, 2, 3, you can see the plus sign as well as the negative sign. The plus sign indicate the growth and negative sign indicate no growth. And in the result part you will find that there is fail, pass or pass. Now here, if there is a positive in all the challenge number, then the disinfectants is failed. But if there is a negative or no growth is found in challenge number 1 or in challenge number 2, then the disinfectants pass the experiment or the disinfectants pass. When the concentration is increased to 2.0, you can see when the challenge number is 1, you will not see any growth of the bacterial cells. In challenge number 2 also, you will not find any bacterial growth. In challenge number 3, only one growth you will find. That means the disinfectant pass. Minimum inhibitory concentration, or commonly it is known as MIC. It gives bacteriostatics values of a test substances or test antimicrobials. The MIC of an antibacterial agents or an antimicrobial agent for a particular microorganism is the minimum or lowest concentration that just prevent the growth of that microorganism. The MIC of a test material is carried out by liquid dilution method or by solid dilution method. MIC determination by liquid dilution method. The step 1, the tube dilutions were made. That means the antimicrobials agents of different concentrations as well as the microbial broth is maintained to make the dilutions. Step 2, the cultures were incubated for 24 hours at 37 degrees centigrade. And in the step 3, the MIC is determined based on the turbidity. Now you can see in the figure that at a concentration of 83.3%, there is no growth of bacterial cells. That means the MIC of the disinfectant is 83.3%. In the tube number 6, you can find the no growth, but in the tube number 5, you can find the growth. That means the MIC of disinfectants is 83.3%. Minimum inhibitory concentration by solid dilution method. The solid dilution method is invented by Cook in the year 1954. In solid dilution method, the nutrient agar is used as a solid medium. Dilutions of antimicrobials or disinfectants are mixed with molten nutrient agar and poured into petri dish. After solidification and dry, a drop of inoculum is added. The advantages of this method are several different microorganisms can be tested on each plate. Precipitation of the medium does not interfere with the result like liquid dilution method. Contamination of a culture plate can be easily detected because of colony features on the solid media are more distinctive. Proving test for new disinfectants. When a new disinfectant is marketed, the following test or following conclusions has to be made. Several test microorganisms like Staphylococcus aureus, Pseudomonas aeruginosa, E. coli are recommended. Test on several temperatures, that is contact temperature and growth temperatures. A disinfection time of up to 30 minutes. Neutralization of disinfectants to avoid inhibitions in the recovery tube. Inclusion of organic matter like proteins or dead yeast cell. After reaction and neutralization, the samples are diluted to give 30 to 300 microorganisms in an agar plate. The other tests include a phenol coefficient test either by suspension method or by capacity test method, MIC, that is minimum inhibitory concentration, and toxicity test like acute and chronic toxicity test is needed for new disinfectants. So students, let us summarize what we have learnt in this module. Specified strains of Staphylococcus aureus, Pseudomonas aeruginosa, Proteus vulgaris and E. coli are usually recommended and obtained from specified culture banks like ATCC for the assay process. Genetic stability of the microorganisms must be controlled in order to prevent the development of a resistant strain. The number of microbes in the inoculums must be uniform under different test conditions. The medium used in the test must be standardized because its composition influences the growth rate and development of a specific metabolite pathway. Strict aseptic condition must be maintained during culture condition and during medium preparations. In diffusion assay, maximum precision results is obtained when medium and assay solutions have same pH. Therefore, the buffer must be chosen carefully. The number of dead and live cells in the inoculums must be controlled for depletion of assay substances. Diffusion of chemical substances before incubation is essential for better and accurate result. Incubation conditions must be uniform during test procedure for accurate result. Thank you.